Hi there guys, and welcome to another A-level biology video. It's Mr. Phillips, and today we are looking at diabetes mellitus, which is the proper name for diabetes. So we need to understand then what diabetes is. Well, diabetes is obviously a disease, diabetes mellitus. There are lots of types of diabetes, but this is the main one, obviously. And about 1% of the people in the Western world suffer from it. And obviously, it's basically causes high blood pressure, high blood glucose, sorry, which can be linked to blood pressure. And there are two forms of diabetes: type 1, which is called insulin dependent or juvenile onset diabetes, because you get it as a child generally. And you are insulin dependent, you're dependent on injections of insulin. And then there's non-insulin dependent diabetes, which tends to appear later in life. And we'll talk about those. Okay, so this is when diabetes is when you're unable to control your blood sugar levels effectively. It can lead to blood sugar levels being too high or too low. High is hyperglycemia and low is hyperglycemia. After a meal, the blood glucose levels might rise, but you haven't got enough insulin or they're being made by the pancreas to tell the liver cells and the muscle cells to absorb, to allow that in, and the permeability doesn't happen. Therefore, the glucose isn't converted to glycogen, and therefore one of the symptoms might be you lose weight, and that is because instead of respiring glucose, which is being instead of glycogen being able to be converted back into glucose, and then you respiring that, you have to respire proteins and fats. Another example is thirst, okay, and that is because the water potential of your blood, it should actually probably say they're too low actually, is lower, okay, because you've got so much sugar in it, and therefore water is needed to dilute it, so it goes away from your cells and into your blood, and that's why you feel thirsty. And glucose in your urine, you're unable to reabsorb high enough levels, such high levels of glucose, so therefore you start losing it in your urine. So here you can see a graph that shows in between meals of someone who's diabetic, someone who's not diabetic. And basically, if you look at the red line first of all, sorry, if we look at the yellow line, we'll go for first of all, you've got someone who is what they say normal, so a non-diabetic person here, okay, and their blood sugar levels after the meal go up, but then they go back down because insulin is released, and the blue line shows when the insulin is released. But the red line shows you a diabetic, so their blood sugar levels are higher anyway, and then after the meal they go up a lot higher upwards, and they last a lot longer because your insulin isn't there to break it down. The only way you get to lose that glucose is by respiring it naturally, because you're not producing the insulin to tell your liver or muscle cells to absorb it. So there are two types of diabetes. The first type is type 1 or insulin dependent diabetes. This is when your pancreas is being capable of secreting enough insulin or no insulin it might release at some point. And obviously, as a result, therefore, your blood sugar levels cannot be controlled. This might be in some cases due to genetics. Again, it might be due to a deficiency for the gene that codes for insulin. But more often, it's the beta cells seem to be attacked by the person's immune system, and there can be genetic tendency for that to happen as well. Type 2 diabetes is non-insulin dependent, okay, and that is when basically not enough insulin is possibly released, but that more so the insulin receptor sites on the cell surface membranes of the liver, the muscle cells, and the fat cells fail to recognize insulin, so they won't respond to it. And that can be brought on by the following risk factors. So if you're obese, you've got a greater chance of being diabetic if you don't do any exercise, high sugar diet, a family history of it, and the older you are, you're more likely to be diabetic. So as the population gets older, you see more and more diabetics. Now, in terms of what can happen, you can get both cases, you get hyperglycemia, which is when instead of having 90 milligrams per 100 centimeters cubed of blood, you can have over 250, which is extremely high. And obviously, your blood levels stay high, and afterwards, your kidneys can't reabsorb all the glucose and something called selective reabsorption, and therefore it's lost in the urine. And it can cause death if not treated, because it can cause all sorts of chaos in your cells, osmosis in places, high blood pressure in places you don't need it. It can damage organs as a result. Hypoglycemia is at the other side of that. Because you haven't stored the glucose away as glycogen, after a meal, you might get very low blood sugar levels. And you do get mild forms of this in normal, healthy people, but it can completely alter behavior, cause tiredness, and obviously it can lead to death if you don't have enough sugar in your body. So in terms of treatments for the two types of diabetes, type 1 The treatment is injection of insulin shortly before meals, but people who are diabetic have to be careful with that because you need to make sure you eat after taking the insulin. Otherwise, your blood sugar level will drop and you'd suffer from hypoglycemia very quickly afterwards. So they have to be very careful. They make sure they do eat. Type 2 diabetics, there is non-insulin dependent type 2 diabetics, and this is where not enough insulin is released, or the insulin receptors, as I said, don't respond. So really, they try to treat that by eating and making sure if your insulin receptors aren't responding, to eat a healthy, balanced diet, make people lose weight. Hopefully, that will start the receptors responding more. Regular exercise to use that glucose up, and you can also take glucose lowering medications that might stop the glucose being absorbed in the small intestines. Often later on, people who are type 2 diabetic will also need insulin injections because if their liver's responding a slight bit and they're not releasing any, that will also need those injections. The source of insulin, so where do we get insulin? Well, it used to be from slaughtered pigs and cows, but there was problems with that, as we'll talk about in a second. But nowadays, it's genetically engineered, which you'll do something about in year 13. But insulin for pigs and cows obviously differed from humans. We don't say it rejects it because it's not an organ, but it would have created an autoimmune response, and that stops it working as well. There's less chance of becoming tolerant to it. So basically, what we mean by that is if we have genetic engineering, it's human insulin you're injecting, whereas our body might actually stop responding to animal like pig or cow insulin as you inject it. There's a more rapid response to genetically engineered human insulin. There's a shorter duration of response, it doesn't last long, which is a good thing, and there's not many ethical issues, whereas the pigs and cows being slaughtered, obviously some people would not be happy. Now, if you're testing for diabetes, what you would do is using the Benedict's test. So you should know what the Benedict's test is. It's where you add the sugar solution, you put it in a water bath for 5 minutes, and the sugar solution will change the Benedict's. It will reduce the Benedict's effectively, and it will go from a blue solution all the way to brick red with high sugar concentrations. The urine should be blue because there should be virtually no glucose in your urine. But this is obviously a way that we can test urine or blood samples for blood sugar. Now, what happens is then you carry out the Benedict's test, okay, which you can look up in your book, but then we're not interested in that. So this is a required practical that you'll do in year 13. We're not interested in the actual red precipitate. We're interested in what's left. So if you have a look at the picture at the bottom, you can see that the less concentrated, so the more concentrated the glucose, the less blue you have at the end of the experiment. So all of the blue here you would get converted into red precipitate. So you'd have loads of red precipitate here. You'd have had very little red precipitate over here at the low concentrations. So because it all got converted into red precipitate, that red precipitate has been filtered out and it is in the filter, leaving just unreacted Benedict's solution. So the higher the concentration of glucose, the lower the blue concentration that is left in the Benedict's solution after the experiment. And it's that filtrate of blue Benedict solution that we're going to test because we can set it up and we can work out the concentration based on how much blue is left in there. There's lots of blue, high amounts of blue, then you will have a low concentration. Okay, so if you have a look at this graph, there you go. And if you've got high concentration, then you have got less blue left over. And that is shown by what we call the absorbance, because lower absorbance will happen when there's less blue and it's more easier, clear, less blue, I suppose. So how would you do this? Well, what you would do is you need to first of all set up a serial dilution of known concentrations. So if we're going to test an unknown concentration, we need to be able to compare it to known concentrations to start off with. So what we would do is in this case we want 4 millimoles of glucose solution. Is you would take that solution, okay, and then you would say in this example they've said 2 milliliters, which isn't a lot, but we'll use these numbers. So they're saying add 2 milliliters of the glucose solution into this test tube. So imagine that green line represents 2 milliliters of glucose solution, and that is at 4 millimolars. Okay, so imagine that is 4. Okay, ignore the numbers below. I'm then going to convert half of that. I'm going to transfer half of that 2 millimolars, so 1 millimolar, into another test tube that's already got 1 milliliter of distilled water. So I've done it with different numbers at the top. I've said 10 and 5 at the top. I'll use 1 milliliters in here. So I already got 1 milliliter distilled water. I then add 1 milliliter of sugar in there. I'm going to halve the concentration because I've got half water, half sugar. So that's then going to drop to 2 millimolars. Okay. I then do the same and transfer another 1 millimolar of that solution in to 1 millimolar of water. Okay, so the key thing is using the same volume of water and the same volume of sugar. That is then going to dilute that down to 1 millimolar, and then I'm going to do the same, mix that solution, transfer 1 milliliter, and then I'm going to add that to 1 milliliter water, and that is going to then again dilute that to 0.5. And then so on and so on. Now you need to make sure, and this is in your AS textbook and it's in the A2 textbook as well, that you could actually explain that process. So one of the questions I've asked is you to explain it as a result. Okay then, so then once we've done that, we've got known concentrations. I'll do the Benedict's test. I'll filter off the brick red precipitate, leaving that blue Benedict solution. And just to remind you again, the higher the glucose concentration, the lower the blue concentration of Benedict's is going to be left over. So therefore, the higher the concentration, the lower the absorbance in a colorimeter. I would then put the volumes of equal volumes of filtrate, so the blue filtrate, into a cuvette. And if you remember, a cuvette is like one of these here, which is like a square test tube that fits in the colorimeter. I would then put it on a red filter. Now, you don't need to know the ins and outs of why that is the case, but it is generally a red filter is what we will use at A-level biology, and it's always worth a mark. I would then put in a cuvette with distilled water in there to calibrate the absorbance down to 0. And then I would put my known concentrations in and plot something called calibration curve. The calibration curve, I would plot my known concentration. So you can't see them that well, but it's on here. But if you imagine that's a known concentration, that's a known concentration, that's a known concentration. These are all the concentrations that I know exactly what those concentrations are, but I'm just plotting them to find out what is their absorption basically at this level. Okay, and so I've done all these different concentrations, and then what I would do is I would then do my unknown sample. I would realize my unknown sample in this case has got this much absorption. Okay, I would draw a line across and I would see, right, okay, so it's just above 3, so maybe 3.2% glucose concentration in this graph. And you need to be able to describe how you would plot a calibration curve. So look what I've written there. You plot the known concentration of glucose on the x-axis, the percentage absorption on the y-axis. I would then plot all my known concentrations before drawing a smooth line best fit, and then I'd test my unknown sample and use the calibration curve to find out the concentration by reading off my absorbance level. Now you can see here type 2 diabetes is on the increase, so you could get something like a question like this, where you can see the blue line is the diabetes, and diabetes between these years, which is 2006 to 2010, has increased pretty much year on year in prevalence, whereas if you look at the green bar, which is coronary heart disease, that has very steadily declined over a few years, and then the other diseases again have declined or just very slightly increased, but nothing in consideration to the change in prevalence of diabetes. So how are we responding to this as healthy and government respond to this? Well, how they're responding to it is by trying to change people's habits. So there's certain things that have happened. So the NHS brought out this Change for Life thing, which is the idea of using a website and using information to help people realise how much sugar is in the food that they eat, and also try and make people make healthy changes such as exercise and stuff like that. That didn't massively make a huge difference, I don't think, on its own. So they introduced then the sugar tax, okay, which is this bit here, and basically for high sugar content foods, they introduced a tax to the foods to try and decrease the amount of sugar people are consuming without even thinking about it. Particularly sugar, that only really affects sugary drinks, the sugar tax, not food, I should have said. And that's because you don't realize you're drinking so much sugar when it's in a drink. So stuff like Coca-Cola, now you see much more Coke Zero as opposed to classic Coke in the supermarkets. And that is as a result, obviously, people tending to buy more Coke Zero these days. Companies themselves have been sort of pressurized into trying to make healthy changes. So stuff like Coke Zero and things like that will have sweeteners in it, as opposed to glucose. Now, there is arguments whether that's healthy or not, but in terms of energy consumption and amount of diet and things like that, it definitely is. And on food, they have put in information now, so you can see this sort of traffic light system of foods. On this example of this burger, saturated fat is high, but obviously there'll be sugars that are high in other foods, and it's just to advise people. So that's on all the food that you can buy nowadays.